everyone. I'm Laura Bush, the editorial director of Spectroscopy, and I'm here today with Micky, Myrick, and Tammi Richardson of the University of South Carolina. Micky and Tammi, thank you for joining us today.
Good to be here.
So you have collaborated in work to characterize the size and taxonomy of phytoplankton using single-cell fluorescence and imaging multivariate optical computing, or IMOC. What was the driver behind this project, and what were your goals?
Well, the driver really was a need within the oceanographic community, and what we needed was an instrument that was capable of detecting phytoplankton in the ocean, and not just detecting it, but characterizing them in terms of what types of phytoplankton are there. But we needed an instrument that is not only really good at doing that, but also has low power requirements. Most of the instruments we put in the ocean need to be plugged in. They need to be tethered to somewhere. And so the goal, I guess the overall goal is for us to work towards developing such an instrument that gives us a really good idea of what types of phytoplankton are in the ocean.
And for this study, why did you choose these two methods, the single-cell fluorescence and the IMOC?
That's really two different questions, and they have different answers. And I'll take that second one first about IMOC. We chose that because it provides the equivalent of a full spectrum measurement very quickly in a very simple package. All the complicated work goes into the construction of the optical elements on the front end, but once that's done, the final instrument has the elegance of simplicity and relatively low power consumption. It's a tool that you might even believe could really be used somewhere in a challenging environment. Instead of just looking at it and going, gee, that's never going to work, which is easy to happen. Another reason that we chose it was that we could. The world's just not bursting at the seams with groups that have the capability of doing that, and we're one of the few that could. The second question, your first question on why we chose single-cell fluorescence excitation spectroscopy as the basis for our interrogation of phytoplankton, let me answer that like this. Phytoplankton species and classes have characteristic light-gathering and photoprotective pigments. And HPLC has been used successfully to identify phytoplankton species by looking at the pigment distribution. Now, HPLC is slow, but fluorescence excitation spectroscopy looks at some of that very same information, and using IMOC particularly, it can be done very quickly. Having said that, we think, and there's some reason to suggest that this is only going to work if you're looking at a pure species and not just some arbitrary mixture of different phytoplankton, which is what you mostly find in the ocean. So in the ocean, you don't get to choose what you're looking at. So we needed some other way to look at purified species, and once you get down to the single cell level, every single cell is a pure species of itself. So presto, we're doing single cell fluorescence excitation IMOC.
Fantastic. And could you explain briefly how it all works?
Sure. IMOC is a technique that uses a series of interference filters. We call them multivariate optical elements, or MOEs, and they tailor the spectrum of the light that's exciting the phytoplankton so the phytoplankton produce distinguishable patterns of fluorescence intensity. It's not usually done with one filter. We usually have a set of filters to do it, and we look for patterns in the fluorescence intensities that come out when you excite a single cell with those different through those different filters. So just as a simple example, you might design and make two different MOEs. Remember, these are interference filters to excite the fluorescence of a phytoplankton cell, and then look at the ratio of the fluorescence intensities you get through those two different filters. And you would have designed those MOEs so that different species of phytoplankton would give distinctly different ratios for classification purposes. So these filters, two or more of them, they go on a spinning filter wheel, and we use a pump to pull water with phytoplankton in it through a flow cell, and that flow cell sits in front of a microscope objective where the phytoplankton get hit with light that's filtered through all those spinning MOEs. And the fluorescence from the phytoplankton is captured as they track across the field of view of the microscope, and then they get lit up by different filters as they're going across, and it's sort of like a barcode. A computer program then goes and finds all those tracks. It finds the individual fluorescent streaks that go with each filter that excited it at that moment. And then we calculate the intensity of fluorescence through those different MOEs, and it presents an array of numbers to another program to be used for classification or for some other purpose. And that's really it.
And how is your approach different from what others have done in this field?
Well, oceanographers have used fluorescence as a way to detect phytoplankton in the ocean for decades now. We go out, we use instruments that excite with blue LED of some sort or filtered light, and we look for the fluorescence, the red fluorescence of chlorophyll A. And the neat thing is that all phytoplankton in the ocean have chlorophyll A, and it's been a really it's something that we've exploited over the years. And more recently, we've moved towards doing spectral fluorescence work where we use multiple excitation wavelengths, and again, looking for that red chlorophyll fluorescence. But the way our approach is different is relates to what Micky's already said about the fact that we have the power of this full spectrum calibration. So the instruments that exist currently rely on maybe 3 or 4 or 5 excitation wavelengths, which means you can really get a limited number of phytoplankton groups, information on those differently pigmented groups, out of the data that you get back. Here we can make filter sets that have really detailed information encoded within them and put those in an instrument. And again, there's the issue of the power. We don't need to have a full-fledged spectral fluorometer to put in the ocean. Really, we're getting that information by using those filters. And again, we want to make something that has very low power requirements, and that way we, in theory, and then this gets a little bit towards what future work that we'd like to do, but if we can make something small enough to fit on a small submarine or maybe a mooring, something that would be deployed in the ocean for a long period of time, that gives oceanographers a lot of information on the different spatial distributions, for example, or how the phytoplankton community in the ocean changes over time. And those are things that really are at the heart of the research interests of ocean ecologists and even climate scientists. I mean, what we do relates directly to phytoplankton grow, they photosynthesize, and they pull carbon dioxide into the ocean from the atmosphere. So they're extremely important both from an ecological point of view. They're food for animals in the ocean, but they're also very important in these biogeochemical cycles. So we're different in that we are combining this full spectrum calibration with the possibility of a very low power requiring instrument that has they're very, very versatile, and they can be used in concert with instruments that already exist.
And what results have you seen so far?
Well, the first thing that we had to do was just to figure out whether this idea was really feasible or not. And the big unknown we had going in was just how variable individual phytoplankton are in an ensemble. So most measurements of fluorescence excitation spectra in phytoplankton are made in bulk culture. So you have thousands or millions of phytoplankton that you're studying at the same time. So it wouldn't be practical to do any kind of single-cell study if it turned out that the characteristic spectra you see in the bulk cultures are only average behaviors and the individuals vary wildly, right? That just wouldn't be suitable for this kind of measurement. So what we needed to do was to gather some single-cell spectra just to design our multivariate optical elements and also to address this question. So to do that, we constructed a single-cell laser trap system to give us detailed fluorescence excitation spectra on individuals. We could hold them for a few minutes and study them over time. And what we found out was that under many conditions, the majority of phytoplankton look very much like the average spectrum of the bulk culture. But there are some exceptions to that, at least one of which occurs when cells are stressed by nutrient limitations. A lot of species show a spectral region that's highly conserved under even those conditions, primarily the region dominated by antenna pigments in that 530 to 610 nanometer spectral range. But there's some cells out there that do change a lot in that spectral window, and we're working on one of those right now as a potential sensor for environmental conditions. So you can see there's 2 or 3 things we learned right there in that introductory study. But there were other results that came out of that introductory work that continue to give tantalizing glimpses of the internal workings of phytoplankton that one of these days we hope we're going to get on to studying. It's one of those things that Tammi and I are postponing until we're retired probably and don't have to go after research funding. But with all those data, we've been able to construct MOEs for phytoplankton classification among a small range of different species, and those appear to work well. We've got a set of papers that are in review at the moment about this that we hope will come out later this year or early in the spring. We've used them out on field trials, and we're still working out how to interpret the data that we get out of field trials, because the field doesn't necessarily have the same species in it that we have in our cultures. And so that's really an unknown element. But we have applied hierarchical cluster analysis and other chemometric tools to the data to show that there are underlying patterns in the responses, and we can associate those with different classes or species of phytoplankton. So there's a sort of a wide-open field for figuring out what's going to happen when you take these into the field and look at real samples and learning how to deal with all that. It's a huge amount of information to try to digest in the gulp. We've also been able to look at the variability in the fluorescence of single phytoplankton, and that's led to some really peculiar observations that I would love to go into these in grinding detail, but I'm going to restrain myself here. We've also shown that the performance of the system can be modeled nicely from its theoretical performance. So this is an unusual system in that we build a complete optical model of every element in it, including the phytoplankton, and that gives us the ability to figure out how it's going to perform in the field before we ever even make the instrument. And that's a real advantage, because then you can build the instrument and have some confidence that it's going to do what you think it's going to do when you put it into the field. And if you happen to be happy with what it's supposed to do, then that's great. Okay. So that's a view of some of the things that we've learned, and we meet every week on this, and we discuss other things that we're learning week by week. But it's been a really exciting project from the standpoint of learning new things for both of us, I think.
Fantastic.
This is the end of Part 1 of using single-cell fluorescence to track phytoplankton communities in the ocean. To hear about what these studies can tell us about ocean ecosystems and the researchers' next steps, listen to Part 2.